Good morning, folks. Wow, what a rainy day we have ahead of us, don't we? So hopefully none of you will have too much trouble with the rain. So please be careful out there. These are unusual circumstances. So welcome back. Let me ask you, as we get started here, do you have any questions after having processed some of the first class periods information and discussion? Anything you would like to ask at this time? Okay, everybody's quiet, so I hope that means that you're doing okay. Let me just take a couple moments here to look at my roster again. Let's see. Do I have a Karen Anderson online? Is Karen Anderson online?
Yes.
Okay, hi Karen. Just wanted to mark you down as having been in attendance. Karen, were you online last class?
Yes, I was.
Okay, thank you. All right. And let's see. Do I have a Virginia Williams online? Is Virginia Williams with us? Okay. All right, gang. Well, again, good morning. You know, it's a tough day to roll out of bed when it's raining like it is, so thank you for being here. I'm going to jump right in. We have to basically cover some chapter 1 today because I want to start chapter 2 after Labor Day. So we're going to jump right in here. There'll be plenty of time for you to ask questions. Of course, we do have lab this morning as well at 9:30, so there'll be time to ask questions as they come up. Okay, I'm going to share my screen with you. And I am going to bring up a Microsoft Word document here. Try to get more of your faces up here so I can see you. Here we go. All right. Okay, gang. So again, as I take notes, I'm going to sometimes refer to pictures and images in your own textbook, and I'll reference that information orally to you. Like, for example, the very first thing I want to talk about is figure 1.1, figure 1.1 in your textbook. I'm not going to bring it up because I don't have a large enough computer monitor that I can show the image and also take notes at the same time. That's one of the drawbacks of this. So I'm going to rely on you to look at your own textbook on your computer when you want to look at things. So we're going to take a look at the levels of structural organization in the human body, which is figure 1.1. Okay. Again, I encourage you to take your own private personal notes as we continue with the conversation. I will take some notes as well. That's what you see on your screen. You know, but if you take your own notes, it forces you to stay focused on the conversation, which is a huge part of staying in the game and being successful in the course. The notes that I do take, I will provide to you. I will put them on Wiley Plus so that you can access them. Okay. So levels of structural organization in the human body, figure 1.1. We need to talk about how material is organized from very tiny material to very large material in the body. And, you know, at first glance, this might seem like a trivial, easy thing, but what I've found over the years, it's often not so trivial. Let me give you an example. If I tell you that a microwave is smaller than an elephant, if I tell you that a microwave is smaller than an elephant, well, that's kind of a silly statement that well, obviously that's true. And you know it's true because you spent your life living in your house and you all have a microwave and you know the average size of a microwave. You've all seen pictures or been to the zoo. You know what an elephant looks like. So yes, a microwave is smaller than an elephant. But when I start talking about things in the realm of biology and human anatomy and physiology, and I ask you, well, Is a mitochondrion larger or smaller than a ribosome? Is a ribosome larger or smaller than a virus? Is an insulin protein larger or smaller than a phospholipid? Well, at that point, you don't have the common reference because you haven't been spending your entire life thinking about these things and being exposed to these things. And what we really want you to do here early in the course is start developing intuition of the scale and relative size of objects compared to each other, because it helps make logical sense of things as you're considering content. So we're going to start a discussion where we're going to start very small. We're going to go small to large here. Okay, and I'm going to bring up a website and go over some ideas with you. And when I'm done with the website, I'm going to come back and put some notes down here on the Word document. Okay. So let's see here. What I'm going to is a website called the Genetic Science Learning Center, which is a fantastic website that's been in existence for a number of years. And let me get my desktop organized here. This is something that you can freely access for yourself. I will put the URL in the chat window for you here in just a moment. Okay, so coming in the chat window is the URL for this website. There you go, if you want to explore it on your own. Okay, lots of interesting topics on this website. Certainly, it's not a requirement. You don't have to do this, but there's topics on human health, right? Precision medicine, neuroscience, all kinds of things to look at. What I'm going to look at this morning with you is under the cell biology section. I'm going to go to amazing cells. Amazing cells. I'm going to click on that, and you can see there's all sorts of interesting things in here. You might want to play with this, particularly as we get into chapters 2 and 3 of the textbook. But for right now, for today's purpose, what I want to talk to you about is cell size and scale. Okay. Now, what I'm going to do is start at the ultra small scale, and get progressively larger. And we're going to talk about things, how they relate to each other in terms of size, physical size. Okay, so, you've all been around for a while, and I'm sure you have heard of the term atom, right? Here's a carbon atom represented on the screen in front of you. You also know, because, you've been alive here, that there are certainly objects that are smaller than atoms. I'm sure you've heard of electrons and probably protons and neutrons, okay? But for our purposes in anatomy and physiology, starting with the atom is a pretty good starting point. So we're going to start here. So if we talk about ultra small things, a carbon atom is a logical place to start, because you realize that just about every molecule in your body that is not water, just about every molecule in your body that is not water is a molecule that is made of carbon atoms. In fact, there's this course that some of you may have to take someday called organic chemistry. And organic chemistry is the study of carbon atoms and the molecules that are made from the carbon atoms, okay? So we have a carbon atom here that's 340 pm wide. In other words, the diameter across the carbon atom is 340 pm. Can somebody please unmute their microphone and tell me what pm stands for?
Is it picometers?
It is picometers. Thank you, Raquel. Raquel, I just wanted to be sure that you were looking at the screen in front of you. Right down here in the lower right corner is picometer. And you can see that a picometer is a trillionth of a meter. A trillionth of a meter. Holy cow, that sounds pretty small, and it is very small. Let me ask a question in the chat window. If I could type properly, this would go better. Here we go. How many inches are in 1 meter? How many inches are in 1 meter? Please type in if you know. How many inches are in 1 meter? Come on, folks. It's okay to be wrong. It's okay to be wrong. I just need you to participate. Okay. All right. Okay. All right. Yeah. Okay. So, so my suspicion is that some of you Googled that. That's my suspicion. Okay. Because I doubt very many of you know that 39.37 inches fit into 1 meter. Okay. And that's fine. Okay. So, let's say that, let's just make it simple at this point. We'll say that 1 meter is 39 inches long. And that's important to know. I mean, in America, in the United States, when I ask that question, typically I get 36 inches because people are remembering their education about a yardstick, right? 1 yard. 1 yard is 36 inches. 1 meter is 39 inches. Okay. So imagine for a moment, if the distance between my hands on the screen that you're looking at, imagine for a moment that that's 1 meter. I don't know that it is because I don't have a meter stick, but imagine that the distance between my hands is 1 meter. It's 39 inches long. Now, if you were to take that distance and chop it up into 1 trillion evenly divided segments, one of those segments would be 1 picometer. So if I took 340 of those tiny segments together, that would be the distance across a carbon atom. 340 picometers is the diameter of a carbon atom. Am I going to ask you to state that on an exam that a carbon atom has a diameter of 340 picometers? No, I'm not. But what I am going to ask you to know is that a carbon atom is smaller than a water molecule. Okay, I'm going to ask you to develop an intuition where you compare the size of things. Okay. All right. So we have a carbon atom, and now we're going to get a little bit bigger. We're going to get a little bit bigger as I move the slide here to the left. Okay. So let me get a couple more things on the screen for us. Okay. I'll start here with the water molecule. Okay. You see that a water molecule is actually 275 picometers. And that's because the atoms of the water molecule overlap each other. So a water molecule, believe it or not, actually is a tiny bit smaller than an individual carbon atom. Think about that, okay? Now, generally speaking, the relationship that we consider here is that atoms are smaller than molecules. That's a pretty logically reasonable statement. But there are very tiny molecules like water that, because the atoms kind of overlap, their overall distance across the entire molecule can, in some cases, actually be smaller than individual atoms, okay? So we have a carbon atom at 340 picometers. We have a water molecule at 275 picometers, okay? Let me ask you this. In the chat window, what percentage of your body weight is due to the percentage of water? What percentage of body weight is due to the presence of water? Yeah, I mean, 80% is a little high. 80% is a little high, okay? But on average, and it's going to vary depending on your personal health status and your current amount of hydration in your body, but you can think about 65 to 70% of your body weight being water weight, meaning that when you step on your bathroom scale, and let's say, just to make the math nice and simple, let's say you weigh 100 pounds, well, 65 to 70 pounds of that 100 pounds is going to be simply due to the presence of water. Everything else is going to be organic macromolecules, which will be a conversation we have in chapter 2, okay? All right, so we have a carbon atom, which is 340 picometers, an individual atom. We have a tiny molecule like water that, because the atoms actually overlap each other, the overall diameter is actually a little bit smaller than a single carbon atom. But then we quickly get to much larger molecules. Okay, let me bring these up. Okay, so here's glucose. Glucose is 900 picometers. So you can see that glucose is a molecule. It's got an average diameter of 900 picometers. That's clearly larger than a carbon atom. So the molecule is larger than the atom. Let me ask you in the chat window, what category of organic macromolecules does glucose belong to? What category of organic macromolecules does glucose belong to? Okay, if you don't know, that's okay. That's why we're here. And certainly in chapter 2, we're going to be really hitting this pretty heavily. Okay, good. Good. Now we're getting some responses. So I noticed the word sugar came up in the chat window, right? Glucose is a type of sugar. It's formerly a monosaccharide, and glucose, being a sugar, being a monosaccharide, fits into the category of carbohydrates. So very nicely done, right? Glucose is a type of carbohydrate. And of course, you probably realize that carbohydrate is associated with energy production in your body. Okay, we have methionine here. Methionine is an amino acid, and you can see its dimensions here. Length and width, 1100 by 700 picometers. Okay, so let's do this. Okay, so the methionine is an amino acid. The question in the chat window is this: what type of molecule inside cells is made of amino acids? What type of molecule inside cells is made of amino acids? What do you think, folks? I'm just trying to get a feel for your background, you know, what your bio knowledge is, okay? So I thank you for all that are contributing a response. It's awesome. I want you to contribute. Don't be afraid to be wrong. Just participate, okay? So yeah, we're getting lots of answers here. It does turn out that proteins is correct, right? Methionine is one of the 20 amino acids. Amino acids are the building blocks of proteins. Nicely done. Okay, so we have glucose, which is larger than a carbon atom. We have methionine, which is a molecule. It's an amino acid larger than carbon atom. Okay, for those that are really basic in their knowledge, you appreciate that a molecule is a group of atoms that stay connected by chemical bonds. This will be a major talking point for us in chapter 2 coming up next week. We have adenine here in the lower right corner. This is a nucleotide. Okay, nucleotides are part of nucleic acids inside cells. They're part of nucleic acids. So I want to ask a question in the chat window. Here it comes. What are the two major types of nucleic acids found inside cells? What are the two major types of nucleic acids found inside cells? Okay, Noah was first on the spot with that. Nicely done. No, it's DNA and RNA. Okay, I'm sure you've heard of DNA and you've probably heard of RNA. Okay, here's another question. Okay. What type of nucleic acid is found inside the coronavirus? What type of nucleic acid is found inside the coronavirus? Right, because we're all online right now because of this darn coronavirus. Okay, good. All right, good folks. It is RNA. That's right. Coronavirus has a genome. It has a genetic structure which is built from RNA. Okay, good job. All right, So the general relationship here is that atoms are the smallest things. You can sometimes have molecules that are even tinier because the atoms overlap, but by and large, most molecules are significantly larger than individual atoms. That's the relationship we would want you to appreciate. Okay, do you have any questions right now? Okay. Let's continue here. A bit more of conversation pieces here. Okay, here we have some phospholipids. Phospholipids form the plasma membrane of the cell. That'll be a talking point in chapter 3 when we get there. Here we have hemoglobin. Hemoglobin is 6.5 nmy. The diameter across hemoglobin is 6.5 nanometers. So here's a question coming in the chat window for you. Is a nanometer larger, or supposed to say, or is a nanometer larger or smaller than a picometer? Okay, good job, gang. Good job. Absolutely, right? Absolutely. A nanometer, as you read at the bottom, is a billionth of a meter. Now, folks, am I going to ask you to know this math stuff for class? Drum roll. No, I won't. But I promise you, if you're going to become a nurse, there will be a moment in your education where you do need to know this math stuff, where you will be expected to calculate and compute with it, okay? So being familiar with it is not a bad idea, because in one of your classes in the future, you will be responsible for knowing these things, okay? All right, so hemoglobin. Can anybody unmute their microphone and tell me what hemoglobin does in the body? What does hemoglobin do in the body?
It has something to do with blood? Does it transport blood, or does it?
Hemoglobin transports oxygen around the body.
Okay, Noah and
I
Scarlett,
think.
yes, you both contributed, and I appreciate that. Noah, you were right. It has something to do with blood. Scarlett, you were right. It has something to do with oxygen. Hemoglobin is a compound, and when I say compound, that means something very precise in chemistry. A compound is a molecule made of at least two different things. And we'll talk about that later on. But hemoglobin is a compound, which is a type of molecule, and it is responsible for binding oxygen or alternatively carbon dioxide. Oxygen or carbon dioxide, and delivering it to the tissues of the body, either into the tissues or out of the tissues, as it were, right? We want to get the CO2 out of our body, and we do that by removing the carbon dioxide from our organs like our livers and moving it to the lungs where we exhale it, okay? So hemoglobin is a gas transport compound. It's a gas transport molecule, okay? This little thing here, 6.5 nanometers hemoglobin, estimates are that hemoglobin is traditionally found predominantly inside red blood cells. Okay, so it's found inside red blood cells, and each individual red blood cell on average contains 280 million hemoglobin molecules. Think about that. Each individual red blood cell on average contains 280 million hemoglobin molecules. Okay, think about it. Could you put 280 million socks in your dresser at home? That would be really hard to pack it in there, wouldn't it? Well, somehow Mother Nature has found a way to pack 280 million hemoglobin molecules inside an individual red blood cell. Okay. So we have hemoglobin here. We also have tRNA here, right? You see that the average length, we're going to be holding one characteristic here, the average one dimension. The average length of tRNA is 7 nanometers. So question in the chat window. What process inside a cell requires tRNA? What does the cell use tRNA for? Okay, well, okay, so I'm seeing replication and production. Production's an interesting word. It's a bit generic. Replication. So I could say, I think T cell transport. Okay. I don't believe so, Jordan, but that was a nice idea. Okay. tRNA is needed to manufacture protein. tRNA is needed to manufacture protein. Okay. So that's what it's used for. Continuing with our diagram here, we have antibodies, right? Antibodies. Right now, if you are vaccinated for COVID, for the coronavirus, your body has manufactured antibodies to protect you. On average, the average diameter across the molecule would be 12 nanometers, okay? All right. So once again, we've started ultra small with the carbon atom and established a relationship where most molecules, with some exceptions like water, most molecules are quite a bit larger than individual atoms, okay? And we're getting even larger here. All right, so next we come up with ribosome. A ribosome is the machine that makes the protein. Ribosomes make protein. Here's a question coming in the chat window. What is the process of protein synthesis called? When cells use their ribosomes to manufacture protein, what is the protein production process called? Okay, well, you will learn if you don't know it. It is called, good job, Kelly. It's called translation. That's right, it's called translation. Okay, right now I'm throwing out a bunch of questions with a lot of odd factoids, right? A lot of odd facts, right? All of this is going to start coming together as we get deep into chapters 2 and 3 of the course. Okay, so if you're confused why I would expect you to know this, well, I don't expect you to know it now, but I will expect you to know it for exam 1. Okay. All right, so ribosomes make our protein, and take a look, right? A ribosome is as big as some viruses, like the rhinovirus, right? The rhinovirus is one of many viruses that cause typical cold symptoms, right? Rhinovirus has a diameter of 30 nanometers. Here we have hepatitis virus. That has a diameter of 45 nanometers. It's okay, Kathy. That's quite okay. Thanks for the message. So when we start talking about atoms and molecules and cells, we appreciate that, you know, most typical viruses are going to certainly be larger than atoms. They're going to be larger than many molecules, and they're going to be tiny enough to fit inside cells. Okay, that just seems logically that we should consider that because we know viruses infect our cells. All right, so let's get a little bit bigger here. Okay, here we have HIV. That's 130 nanometers wide. We have the influenza virus. That's 130 nanometers wide. Coronavirus, which we're dealing with in society at this point. Coronavirus has a diameter of roughly 100 nanometers. Coronavirus particles are about 100 nanometers wide, okay? A coated vesicle, well, that would refer to something that is exported from a cell, transported through the bloodstream, and delivered to another cell. So cells actually communicate in your body by ejecting these little bubbles that float through the bloodstream and bind to a target destination, another tissue, another organ, and then those coated vesicles are brought into the cell by processes known as endocytosis. Okay. All right, so atoms, molecules, bigger, bigger, bigger. Okay. Here we go. All right, so we have a measles virus there, 220 nanometers long. All right, now we're getting to some things here. Okay, we have a mitochondrion. That's, you see, 4 by 0.8 micrometers, 4 micrometers long. So you notice in our conversation, we've gone from picometer, which is PM, to nanometer, which is NM. I now have micrometer, which is kind of like UM. It's not really U, it's the Greek letter mu, but micrometer. 1 micrometer is a millionth of a meter. 1 micrometer is a millionth of a meter. Okay, so I want to show you something kind of strange here. Take a look. E. coli bacterium. An E. coli bacterium is 3 micrometers long on average, and a mitochondrion is 4 micrometers long on average. Okay, so let me ask a couple questions here in the chat window. True or false? Mitochondria are found inside human cells. That's definitely a true statement, right? Mitochondria is a type of organelle. It's a membrane-bound structure that's responsible for producing biochemical energy for our cells. That's definitely true. Here's another question. True or false? E. coli bacterium are normally found inside human cells. Okay, good. I'm very happy with responses. It seems like many of you have a certain level of background and intuition here. Right, E. coli is not normally found inside our cells. Here's another question. Based on the size of E. coli, could it fit inside our human cells? Okay, you see the logical conclusion here, right? Mitochondria are found inside our cells. E. coli bacterium are not normally found inside our cells, but because the E. coli bacterium is basically the same size as a mitochondrion, from a physical size perspective, it could fit into our cells. And that's one thing that we talk with students about in microbiology, because we all know that viruses infect our cells, but what is much less commonly known is that there are many species of bacterial pathogens that do enter our cells and cause trouble inside our cells. Okay, so let me ask you a question. Have you heard of a theory called the endosymbiotic theory? Okay, some of you say no, some say yes. Folks, I'll give it to you in a nutshell so we can move forward. The endosymbiotic theory is this. Many scientists propose, and there's pretty reasonable evidence for this, that the mitochondrion on your screen, the mitochondrion, which is a normal part of your cell, is actually an ancient primitive bacterial cell that resides inside our human cells. The evidence seems to suggest that mitochondria are actually primitive bacteria that have merged into our cells along long geologic timescales throughout evolution of life on our planet. Okay, that opens up some intriguing possibilities that we could talk about at another time. Okay, Thoughts or questions?
Yeah, I had a thought. So when I was in LPN school, we took anatomy, but it's not like as focused as this one, I guess, or more or as intense as this one. But they talked about the human biome and how that's like a new thing that they're studying because, like, everybody when antibiotics were first discovered, they were giving antibiotics everybody for everything, and then they started realizing that like antibiotics are good if they're used when they're needed, and that we have actual bacteria that help us that we shouldn't be getting rid of, like in our gut. And and like I remember our teacher telling us about how our bacteria, like for instance in our stomach, actually affects other parts that goes on in our body, and like it could also affect how we feel and stuff like that. Because like I don't really remember all the, but I remember her telling us about the human biome and how that's like a big thing now, and they're trying to study that. And maybe that kind of goes with the mitochondria and how it used to might have been a bacteria and now it's formed a part of our body.
Scarlett, thank you for those comments. And you know what? I basically agree with everything you just said. That was fantastic. Okay, fantastic. So in terms of terminology, it's typically referred to as the microbiome, but you're right, this community and civilization of different microorganisms that live inside and on the surface of our body, which contribute to our overall health, and which we're just at the very beginning of actually understanding how this all works. But you're right, there are bacteria and other microbes in our gut, in our colon, that actually manufacture molecules that travel through the bloodstream up to the brain, cross the blood-brain barrier, enter the brain, and influence our brain chemistry. So therefore influencing how we might feel at any moment of time. Okay, it's fascinating stuff we don't completely understand. Okay, thanks for saying that. All right, let's continue here. We're getting bigger, bigger, bigger. Okay, so we get to baker's yeast. That's 3 by 4 micrometers. Thank God for baker's yeast, right? We enjoy some bread and alcohol. We have an X chromosome, right? The gals have 2 copies of the X chromosome at 7 micrometers long. Guys have 1 copy of the X chromosome. Here we finally get to a human red blood cell, about 8 micrometers in diameter. Okay, about 8 micrometers in diameter. Okay, this is the object that I reported earlier, contains about 280 million copies of hemoglobin. Okay, all right, getting bigger, bigger, bigger. Okay, we have skin cells. Look down at the surface of your arm. You see epithelial cells. Average diameter is going to be 30 micrometers. We see a tiny sperm cell, which is very narrow but long at 60 micrometers long. We see a female egg cell, 130 micrometers wide. Look at the size of the human egg cell compared to the sperm. We also have photoreceptor cells, rod cells. Okay, so let me ask you this in the chat window. Where inside your body do you find rod cells? Okay, gang, very nice. The eyes, right? And specifically, you find the rod cells that are part of the retina at this. It's the back layer of nervous system tissue at the back of the eyeball. Good job. All right. So we're getting a little bit bigger. Here we have a paramecium. We have a paramecium and amoeba proteus, both of which maybe you studied in high school biology. You might have had a chance to look under the microscope, right? Paramecia and amoeba. There are different forms of these things that you can actually find forms of paramecium and amoeba that are pathogens to animals and to humans that can cause harm. Okay, and then we're getting even bigger, bigger, bigger to things that you have more of an everyday life experience with. A grain of salt, right? I'm sure, even if you had some cereal this morning, there was some salt in the cereal, right? There was some sodium chloride, 0.5 millimeters in diameter. We get bigger, bigger, bigger. a sesame seed you might have on your bagel. We get to a grain of rice, which you might have with your lunch this afternoon, 8 by 2.5 millimeters. We get to a coffee bean, 12 by 8 millimeters, right? A millimeter is 1,000th of a meter. So we've gone from 1/trillionth of a meter to 1/billionth of a meter to 1 millionth of a meter to 1,000th of a meter. We've gone from small to large. And this is where our animation, our visual aid runs out. 12-point times regular font that you might use to write your English reports with, okay? So folks, I don't expect you to know the specific numbers and math of any of these things at this point. I don't expect that, but I really do want you to start to develop an intuition of the scale and relative size of objects compared to each other. That's important because it helps you to logically reason about questions, okay? So my plan next is to document some of the things we just talked about in your notes. Do you have any questions? Okay, so we're back on page 3 of your textbook, figure 1.1. I'm starting small to large. So we have atoms. And what I'll just do is put up an example here. Okay, so we had carbon atom. Can somebody please unmute your microphone and tell me the name of any other kind of atom you know about?
Hydrogen.
Hydrogen atom, that's a fantastic one. How about one more?
Oxygen.
Okay, oxygen, good job. Okay, basically, if you remember that horrible thing that you might have had to wrestle with in high school called the periodic table of elements, right? Periodic table of elements tells you all the different types of atoms known to exist to mankind. All right, so we have atoms, which will be our starting point as very, very small objects. Next, we build up to molecules. Okay, and so we did admittedly have a single molecule, water, that was shown to be smaller than, that was shown to be smaller than a carbon atom. But generally speaking, most molecules are larger than atoms. And so let me start with, an example that was in the visual aid that I used, right? We had tRNA. tRNA molecules are molecules. Someone give me another molecule.
Glucose?
Okay, thank you very much. Glucose is a perfect example of that. Okay, glucose is a perfect example. And we mentioned protein. So let's do insulin, right? Insulin is a protein, right? It's a hormone, but it's also a protein. Those would be examples of molecules, okay? All right, so we have atoms, then we go to molecules. Now, if you look at your textbook, it shows atoms, then it's figure 1.1, it shows you atoms, then it goes to molecules like DNA, then it goes to cells. But folks, I think there's something else we need to talk about inside here, which figure 1.1 isn't showing you, but which I did mention as I had the conversation at the visual aid, and that would be nanostructures and nanomachines. Okay. And so by this, I mean things that are larger than molecules, but smaller than cells. Larger than molecules, but smaller than cells. Nanostructures and nanomachines are made of groups of molecules, right? So an atom is a basic building block of matter. If you link atoms together, you typically build larger objects like molecules. Most molecules, 99.9% of molecules, are going to be smaller than atoms. The only exception is when you have molecules whose atoms are overlapping each other, like water, then sometimes you can get a tiny molecule that's smaller than a single atom. But atoms are smaller than molecules. If we link molecules together, we build nanostructures and nanomachines, which are smaller than cells. And these would be things like the organelles of a cell. So that would include mitochondria, right? Mitochondria, lysosomes, nucleus, etc. Okay. It would also include nanomachines, like the best, most famous nanomachine I can quickly give you is a ribosome. Okay, there are certainly other nanomachines. There are things called kinesin and dynein motors inside cells, which move cargo along the side of skeleton. But I'll just say a ribosome at this point for an example. Okay, so molecules are made of groups of atoms. Nanostructures and nanomachines are made of groups of molecules. Okay. Next, we get to the actual cells. Okay. Now, here's where you'll probably start to get more comfortable. Okay. Example of a cell would be a human cell. But can anybody give me an example of any other kind of cell? I mean, you can be more specific than human cell. You could say epithelial cell or something. So, folks, let's get some examples of other cells.
A blood
Yeah.
cell?
Okay, so we could talk about red blood cells, and we could talk about white blood cells. Was there another response?
Fat cells?
Fat cells. Okay, all right. Yeah, you're right. A fat cell, which is technically an adipocyte. Okay, beautiful things. Let me mention one other thing, right? Don't forget about your microorganisms like bacteria. Okay. Truth be told, we won't talk too much about microorganisms in this course because we're focusing on the anatomy of the human body. But when it comes to health and disease circumstances, we really need to talk about microbes, and you'll get more of that kind of conversation typically in a microbiology course. Okay. So, something came into my mind in terms of nanostructures and nanomachines. I think it would be also appropriate to include viruses. Okay. Remember that viruses are not cells. Viruses are not cells. They are not cells. Okay. All right. So atoms to molecules to nanostructures and nanomachines to cells. Okay. We're talking about size and the relative relationship of things. Next, if we group cells together, well, we build tissue, right? Tissues. And we haven't talked about that yet, but there are 4 major tissue categories. Let's list them out. It's epithelial tissue, muscle tissue, connective tissue, and nervous tissue. Now, there are different subcategories of each of these tissue types. For example, muscle tissue, right? Muscle tissue is going to have skeletal muscle tissue, which moves your bones. It's going to have cardiac muscle tissue, which is your heart muscle, which pumps the blood through your body. You also have smooth muscle tissue, which lines blood vessel walls and the walls of cylindrical structures in the GI tract, such as your intestines. Okay, so each of these categories, epithelial, muscle, connective, and nervous, has subdivisions. You will learn all about tissues in chapter 4, and you will learn more than you ever want to know about these things. All right, so we have tissues there after cells. If we link tissues together, obviously we next get to organs. For organs, pick your favorite organ, right? You talk about a heart, liver, kidney, spleen. Believe it or not, your skin is an organ. Believe it or not, every individual bone of your skeleton is an organ. Bones are organs. Bones are organs, okay? So we have that. You're going to be learning the identity of many bones in the lab. It's important to appreciate that each bone is an organ. Okay, if we link the organs together, we get to organ systems. Organ systems. Okay. There are, and if you've started to read chapter 1, you already know the answer to this. There are 11 organ systems in the animal body. There are 11 organ systems in the human body. You see them identified as part of table 1.2 on pages 4, 5, 6, 7. 4, 5, 6, 7. there are 11 organ systems, and I'm not going to list them all out, but just as an example, right? You have the endocrine system, you have the digestive system, nervous system, etc., etc., etc. Okay. All right. And we're almost ready to finish up this topic in that if we link our organ systems together in ways that allow them to communicate each other, which is typically done either by the nervous system, right? Neurons down nerve fibers will communicate between organs, or by molecules distributed through the bloodstream like hormones, you get the entire complete animal body, the complete human body. All right. Okay, gang. So do you have any questions or thoughts right now? Okay, so don't be surprised if a question or two shows up on a future exam where I try to challenge your intuition about the organization of material, okay? I do want you to appreciate this general hierarchy of atoms to molecules to nanostructures and nanomachines, cells to tissues to organ systems to the entire body. Okay, all right, good stuff. Let's continue forward here.
I have a quick question.
Okay,
So in the book, it said that atoms and molecules are part of the chemical structural level. I'm guessing that includes the nanostructures and nanomachines as well. Am I correct with that?
yes, yes. If you wanted to use the organizational detail of figure 1.1, I would put nanostructures and nanomachines in the chemical level, okay? The reality of it is that everything inside your body is made of molecules, okay? Molecules use chemical reactions to accomplish goals and purposes and functions. So talking about atoms, you notice in your textbook between number 1 and number 2, between blue ball 1 and blue ball 2, you have molecule, right? Molecule sort of fits between number 1 and number 2. It would be appropriate to put nanostructures and nanomachines to the right of the molecule diagram you see in figure 1.1.
All right, thank you.
Other questions? Okay. All right, folks. So, there's a lot of things in each chapter. I won't lecture on everything on every chapter because honestly, there's no time. I mean, if I literally lectured on every page of chapter 1 or any chapter in the book, we could take 2 to 3 weeks per chapter, and we just don't have that kind of time. So I will always pick and choose topics that I want to spend lecture time on. It doesn't mean the other things aren't important. It doesn't mean you don't have to read the other things. You do. It doesn't mean I won't test on the other things. I will. Okay, so please understand there's limited lecture time, and I will pick certain topics to focus on. You can always ask me questions about other things you're reading. I can try to help you with things you're not understanding.
Quick question, Doctor, Professor Allen.
Yeah.
For like when we have the exams, are you gonna have like a review paper or anything like that, or just review
Yes.
on our own?
Okay, 2 weeks before every exam, both lecture and lab exam, I will give you study information. For lecture exam, I will give you a study guide that will give you a list of vocab terms that will be very important for you to know. And I will give you some very strong hints about the major essays that will be on the exam. So you can focus your study efforts 2 weeks prior to the exam. So you will get some help there.
Thank you.
Anything else? Okay. The second major talking point that I want to spend class time on, and I'll probably need to spend a little bit more time in lab today, and that's okay, is homeostasis. Okay, page 9 in your textbook starts the topic of homeostasis, and I want to talk to you about homeostasis. Okay. So, homeostasis, just before I get into the technical details, is really kind of talking about the idea of the soup or porridge in the Goldilocks fairy tale, where Goldilocks had porridge and we didn't want it to be too hot or too cold, right? We didn't want the porridge to be too hot or too cold, okay? So homeostasis is talking about that. How do we ensure that our soup is not too hot or too cold? If the soup is too hot, how do we make it colder? If the soup is too cold, how do we make it warmer? Okay, that's basically the essence of homeostasis here, but as applied to the human body. Okay, so I'm looking at chat window. Sometimes, okay, Kelly, I see the question. I see the statement. Thanks for letting me know. Thank you, Israel. That's okay. All right, let's continue here. So, homeostasis as a talking point. First, let's just define this, okay? Let's just define homeostasis. Homeostasis refers to the state of balance, or perhaps you prefer equilibrium, inside the body that is maintained by regulatory mechanisms working inside cells, tissues, organs, and organ systems. Okay, that's kind of a generic starting point definition. Homeostasis refers to the state of equilibrium, the state of balance inside the body that is maintained by regulatory mechanisms, of which we're not defining them yet, that work inside the cells, cells together as tissues, tissues together as organs, organs together as organ systems. Okay, so we're talking about all the machinery inside the cell that can detect and respond to abnormalities in some body parameter. That body parameter might be temperature. I mean, you know this. Okay, so I'm just pausing because I have a chat question. Is there a separate Zoom for lab? No, there is not, right? You will use the same Zoom link you're currently on for lab, okay? All right. So, intuitively, in the summertime when you're outside and it's warm and your body is too hot, what's your body going to do?
Sweat.
It's going to sweat, right? So you need some kind of mechanism that responds to the accumulation of too much heat energy inside your tissues. Okay, so you sweat and cool yourself down. Similarly, in the wintertime, when you're exposed to the cold air and so forth, your body temperature drops. And so you get too cold. How does your body typically respond then?
Goosebumps.
River.
Okay, I'm sorry. Two folks talked at once and I appreciate it. I think you said the right thing. I just couldn't understand, but I think you said shiver, right? So we tend to shiver, right? So we have this repeated cycle of muscle contraction and relaxation. And as our muscles shiver, as they contract and relax, to contract, they have to burn energy. And one of the waste products of burning chemical energy is the production of heat energy, which then warms our bodies up, right? So this is really what we're talking about. We're just going to put some more formal terminology on this. So let me give us a for example here, and I'm going to use a different for example. Okay. So here's an example with blood glucose. concentration, blood glucose concentration. And so I'm going to put a question here. Okay, let me ask you folks, what is normal? When I say normal, that's a loaded word, right? Because what's normal to one person might be not normal to another person. But I'm talking from a statistical average perspective. What is the normal? What is meaning? What is the healthy resting? What is normal, healthy resting blood glucose concentration? Okay. This morning, when you woke up after having slept for 7 or 8 hours, so presumably you didn't eat any food overnight because you were not sleep eating. Okay. All right. So I just popped up the chat window here, and some of you had some very nice responses, right? 70 to 110. Okay. And I love that. I love that because that's basically right. And, you know, depending on what reference you read from, you might get a slightly different range of values. So we won't argue with that. But 70 to 110 seems pretty good. But I'm gonna pick on you and ask you this: what are my units? Because 70 to 110 means nothing. What are my units? Okay. You know, in everyday life, we like to throw numbers out and we assume people really know what it refers to. But is that 70 to 110 apples? Is it 70 to 110 oranges? What is it?
Milligrams? No.
It's okay, Brittany. Don't get worried just
That's
because I go
what I
like
was
this.
doing.
Okay, so Brittany, you are half correct. You are half correct. Help Brittany out. She didn't get the whole thing, but she got us halfway there. 70 to 110. Brittany said milligrams. That's partly correct. The reason it's partly correct is because I'm talking about concentration. You need to think about what concentration means.
Milligrams per liter?
Okay. Now, Raquel, you're not technically correct, but you got me to the other part of my unit answer. Okay, a concentration is an amount of substance, such as milligrams, dissolved in a volume of liquid. Okay, now it's not liter, but it's deciliter. So this would be milligrams of glucose per deciliter of blood plasma. 70 to 110 milligrams of glucose per deciliter of blood plasma is the units of concentration for resting blood glucose. Okay, so, all right. Okay, so folks, I want to show you something about your own textbook. Okay, I want you to see that your own textbook has some very useful reference information in it. If you would please, and I know you don't have a physical textbook, but you need to go to the back of your digital textbook and look for Appendix C as in Charlie. Look for Appendix C as in Charlie. Appendix C as in Charlie should have normal values for selected blood tests. Normal values for selected blood tests. And so just to see if you're finding the right page, please find Appendix C in your textbook and please tell me what the normal, now let me see here, tell me what the normal total iron concentration should be in males. Type it into the chat window. What is the total serum iron concentration supposed to be for males? If you've been able to find Appendix C, you should see it there. Total iron concentration in males. What's normal reference value for males? Okay, there we go. We're starting to get it. Okay, I'm not asking that because it's immediately important that you memorize it. That's not the point. I want to know that you're accessing information in your own textbook. That's what I'm interested in. Okay, so we're starting to get correct answers. Hopefully everybody can ultimately find this Appendix C. And that's the point, that there is useful reference information in your book that can be very helpful. You don't have to memorize these things. That's why we have books. That's why we have tables. That's why we have computer databases. That's why we have smartphone applications. Okay. All right. Good job. Good job. Okay. Let's get back to the conversation here. So the normal resting blood glucose concentration is 70 to 110 milligrams per deciliter, or more completely, milligrams of glucose per deciliter of blood plasma. Okay. All right. So, so how will the body respond if blood sugar is too high? How will the body respond if blood sugar is too high?
You'll get dizzy, pass out, or feel lightheaded.
Well, okay, maybe I need to be more precise with my question. If it gets too high, the symptoms you just stated, Brittany, is typically characteristic if it's too low.
Too low.
Okay, if it's too high, you may not have any symptoms. You could go into shock and death. But I guess what I'm trying to get at is, if your blood sugar is too high, your body will experience some symptoms, but how will your body respond to counteract the blood sugar being too high? How will it respond? Not what will it experience in terms of symptoms, but how will your body respond?
He'll try to correct the insulin production rate in the body.
Okay, so I heard the word insulin, Shay, and that's fantastic. Okay, it will try and produce more insulin. That's true, Jordan. You appreciate that the production of insulin is something that takes time. It doesn't happen instantaneously. Okay, so if you do have blood sugar that's too high, your pancreas will begin the process of making more insulin. But even before that, or hopefully to immediately take care of the localized emergency, your pancreas will release the insulin that it has already made and stored, right? So we will release insulin. And this is not yet a lecture on endocrinology. That's something we can do later, and honestly, you'll get that in AMP2. But the insulin floats into the bloodstream, and then the insulin promotes certain target organs and tissues of your body to respond by drawing glucose inside the cells. Okay, so when insulin's in the bloodstream, it will bind to skeletal muscle cells and liver cells and cause those cells to bring glucose into the interior of those tissues. And by doing that, we lower the blood sugar. Okay. All right. So question, how will the body respond if blood sugar is too low? Again, I'm not asking about the symptoms. I'm not asking about the fact that you might get shaky. I'm asking, how will the body try to counteract blood sugar being too low? See, everybody knows about diabetes, right? So you treat diabetics with insulin, right? But what about if it's the opposite circumstance?
Does the body start releasing glucagon?
It does, Scarla. Nicely done. The body will release glucagon, which is also a hormone produced and released from the pancreas. And the purpose of glucagon is to stimulate the production of glucose in your cells. In other words, your body is a magnificent machinery made of nano machines, and it does have the capacity, if it's given enough time and resources, the chemical machinery of your cells can literally stitch together carbon atoms into glucose molecules in a process called gluconeogenesis. Okay, so let me just stay here. Up top, release insulin to lower blood sugar. And down below, release insulin to elevate blood sugar, not insulin, sorry, glucagon. Release glucagon to elevate blood sugar through gluconeogenesis. Okay. All right. So I think, folks, are you feeling okay right now with this conversation? Okay, Rachel, I see about turning stored glucose into glucose. Okay. Rachel, you say in the chat window, and I think this was an answer to the second question, how will the body respond if blood sugar is too low? And you say turn stored glucose into glucose and release it. I like what you're telling me there. I would just correct the language. It will turn glucose that has been stored as glycogen back into glucose and release it. Okay, so we can also add that. So it will release glucagon to elevate blood sugar through gluconeogenesis and also stimulate the conversion of glycogen into glucose. Okay, glycogen is a polymerized version of glucose. By polymerized, I mean it's a chain of glucose molecules that have been linked together to form a chain. So glycogen is a glucose linked to a glucose, linked to a glucose, linked to a glucose. Okay. Okay. Folks, are you feeling okay with this conversation so far? Any questions? Yes.
Just for clarification, when glucose is stored, like anytime it's being stored, it is considered glycogen or glucagon. And then from there, if we want to use it again, it is converted into glycogen.
Yes, I believe you did say that correctly. Just to reaffirm what you stated, anytime you have excess glucose in your body, one of the things your tissues will try to do is try to take the excess glucose inside the cells of the skeletal muscle and liver, and it will link the glucose together into a chain, and that chain is called glycogen. So excess glucose is stored as a chain of glucose called glycogen for future use. That glycogen will be converted back into individual glucose molecules in response to glucagon. I know it's confusing because you got all these G words, right? Glucagon, glycogen, glucose, gluconeogenesis. It gets really confusing. Okay, any other questions? All right, so now that you're getting the basic idea of what homeostasis is all about, all we need to do now is put some formal terminology down. Part of education is using fancy words so you sound sophisticated. And I'm not a big fan of that because, sophistication tends to kind of obscure things. But there is a certain amount of vocabulary we need you to know. Okay, so I'm going to talk about continuing here, the control of homeostasis through feedback systems. Okay, and so you see feedback systems talked about on page 10 of your textbook. So let me define what a feedback system is. Okay, a feedback system refers to all the components of the body. So when I say components of the body, I'm specifically referring to the cells, tissues, organs, organ systems, right? Cells, tissues, organs, organ systems, okay? A feedback system refers to all the components of the body that work as a team to monitor a body condition. Well, what would that body condition be? It could be blood glucose, right? We've just been talking about it. That would be a body condition. It could be blood glucose, okay? It could be blood pH levels. Any of the conditions you saw in Appendix C, Appendix Charlie, are body conditions that can be monitored by the anatomy of your body. Okay, so a feedback system refers to all the components of the body that work as a team to monitor a body condition and which take corrective action to restore those conditions back to healthy, homeostatically normal values when the condition is out of balance. Okay, so let me read that in my notes one more time. I know we got to take a break for lab. A feedback system refers to all the components of the body that work as a team to monitor a body condition and which take corrective action to restore those conditions back to healthy values when the condition is out of balance. That is a feedback system. Folks, it's 9:16. Let's take a break here before we go into our lab time together. Here's what we're going to do. We'll take a break. We'll start lab. I will continue my conversation here about homeostasis for maybe 30 to 45 minutes, half an hour or so. We will finish that. We will be done with chapter 1. Then we will do lab content today that we need to get started with, okay? Folks, I'd like to take a nice break at this time. Let's start at 9:35 for lab. Start at 9:35 for lab, okay? So I'm going to keep this Zoom link open. The Zoom link will stay active and on. You don't need to log off. I'm just going to turn off my video for the next 15 minutes, okay? So go have a nice restroom break, get yourself a cup of coffee. I'll see you at 9:35. Okay, gang, welcome back. I ask you to refocus in so we can get started on our lab time together. And so, okay, what I'm going to do is finish our conversation about homeostasis in hopefully about 30 minutes, and then we'll do some dedicated lab activities. Do you have any questions you'd like to ask right now? Okay, all right, well, please let me know. Remember, the chat window is there. You can always pop a question into the chat window. All right, so we're talking about homeostasis. We just defined feedback systems. So again, we're on pages 10 and 11 of your textbook. And so there's some other terms here we need to define. We need to define the components of the feedback system. Okay, so there's a comment to me on the chat window about whether the notes that I'm typing will be posted. Yes, the notes I'm taking right now to help move class forward will be posted on Wiley Plus in the Files tab, of Wiley Plus. Okay. All right. So major components of the feedback system. Okay. Let's take a look here. Okay. So number one is this idea of receptor. Okay, so let's define it, maybe give an example of it, talk about it a little bit. A receptor is a body structure. Okay, now when we say a body structure, that's a very generic phrase. Okay, what would be an example of a body structure? Well, I could be talking about the pancreas, right? In our previous example of high blood sugar, the pancreas has detected that blood sugar is too high, and so something has to happen. So a receptor is a body structure, a tissue, an organ, or at the most fundamental level, a cell, okay? So a body structure, and that body structure, like an organ, is almost always going to be innervated. There are going to be nerve endings there, right? There's going to be neurons present. So even in the pancreas, right, there are nerves that serve the pancreas organ, right? The vagus nerve communicates between the brain and the pancreas. So It's a body structure that typically includes a nerve, that monitors changes in a body condition. Okay, for example, again, blood sugar, and communicates that information to a control center. Okay, so we're going to talk about the control center in just a moment, but let's talk a bit more about some nuances of this idea of a receptor. So for example, in terms of an example here, when we were talking about blood sugar, okay, the receptor would be cells of the pancreas and, believe it or not, cells in the hypothalamus of the brain. So my first point here is that, we're talking in generic terms, right? Now we're defining a receptor as part of a feedback system. But, in terms of controlling and monitoring a body condition like blood sugar, you can have multiple receptors at multiple anatomical locations of the body. It's not just a single receptor that handles everything, right? The body's much too dynamic for that, okay? So appreciate that the body can have multiple receptors that help measure a body condition. Okay, there can be more than just one receptor. And in this example, blood sugar shares that with you. Okay. All right. Now also, excuse me, letter C. The word receptor is an overloaded term. And what I mean by that is that receptor can have different meanings depending on context. Receptor can have different meanings depending on context. Okay. So, let's see here. Okay. In the current context of our conversation with blood sugar, context of conversation with blood sugar, receptor is referring to the cells of an organ, like the pancreas. Okay, there are going to be cells in the pancreas that measure blood sugar. There are going to be cells in the pancreas that measure blood sugar. And so we would call those cells generically receptors or receptor cells. Okay, that's how we're using that term receptor now. Okay, but there, as we get into more complicated things, you appreciate that the cell itself, the cells I'm referring to here now that are part of the pancreas that are monitoring blood sugar, well, it's not the entire anatomy of the cell that monitors the blood sugar. The cell is not just doing that. It's not just monitoring blood sugar and doing nothing else. So, at the molecular level, receptor refers to a molecule, typically a protein, which binds to a substance like glucose, if we're talking blood sugar. Okay. At the molecular level, receptor refers to a molecule, typically a protein, which binds to a substance like glucose. So what we're really saying here in the context of our conversation is that the receptor for blood sugar are cells that contain proteins on the surface of the plasma membrane. Pancreatic cells that contain proteins on the plasma membrane surface of the pancreatic cell that are capable of binding glucose in some fashion. Okay. Most precisely, the receptor is a protein on the surface of a pancreatic or hypothalamus cell in the brain, right? Because there's at both locations. Most precisely, the receptor is a protein on the surface of a pancreatic or hypothalamic cell that can bind glucose. Okay, so I don't think your book quite goes into this level of nuance in chapter 1. Okay, but I wanted you to appreciate it because the idea of what receptors are is going to come up in other contexts throughout the course, and I want you to appreciate that that word receptor can kind of refer to different things in different contexts. Okay, context is so important in everything in life. Okay, let me ask you a question in the chat window. Here you go. What is the first thing that comes to mind when I state the word Corvette? Okay, car. Okay, fast. Good. All right. Yeah, absolutely. Right. I mean, that's what we think of, you know, whether you like cars or not, whether you're into sports cars and fast cars and blah, 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 doesn't matter. But we think of Corvette as a car. But you appreciate that the term Corvette in another context, in the terms of the Navy, actually refers to a naval vessel, right? A Corvette is a type of naval vessel. And so that's my point. If our context would have been talking about the Navy, then really the first thing you should have thought about when you heard the word Corvette is a specific kind of ship. And that's my point here, is that words, you know, the English language is an amazing thing, but it's often a very subtle, fickle thing in that, you know, the context of the conversation will dictate what the meaning of the word is supposed to be. Courtney, I do see your comment. Okay, is there something, Courtney, you would like me to repeat?
No.
Okay, so am I going too fast in terms of speaking or in terms of typing?
Both.
Both? Oh, okay. Well, it's okay. Sometimes I get a little excited and I want to get through content, but remember, the lecture is recorded and the notes will be provided. So hopefully you're not really missing anything, okay? But feel free to ask questions along the way. Okay, so let's go back to this. A receptor is a body structure that monitors changes in a body condition and communicates that information to a control center. In the current conversation, what do I mean by a receptor? Well, in terms of our blood sugar example, I'm talking about a cell of the pancreas or a cell of the hypothalamus of the brain. And I won't say any more, short, sweet, to the point. Folks, is this making sense? Okay. All right. So let's define what a control center is. Okay, let's see what a control center is. So I should use number 2 here. Control center. Okay. A control center refers to another body structure. Okay. This body structure will typically be the brain, because, you know, when we talk about regulating body physiology, number 1 is the nervous system. The nervous system is the master. It's a very close commander of the body would be the endocrine system, so hormones, okay? But number 1 is nervous system, number 2 is the endocrine system. So when we talk about control center, We're talking about typically the brain, that determines what the range of values should be for a body condition, and which orders other parts of the body to take corrective action when homeostasis has been disrupted. Okay, let me say this again. The control center also refers to a body structure, usually the brain, that determines what the range of values should be for a body condition, right? We stated that resting, fasting blood sugar is about 70 to 110 milligrams per deciliter, and cells in the brain, specifically a cluster of cells in the hypothalamus, are going to monitor that, and they're going to say, oh, this is not quite right. My blood sugar is too low, or my blood sugar is too high, okay? And so the brain is going to communicate to other parts of the body to take corrective action, okay? So, in our example here, cells of the brain, the hypothalamus, not only act as receptors, but also act as a control center, okay? Cells of the brain in the hypothalamus not only act as receptors, but also act as a control center. So this is the next point. Let's see here. Is that sometimes body structures perform double duty. Okay, sometimes body structures perform double duty, which means anatomy can act as both receptor and control center. Okay. All right. So in our example, okay, the receptor for the blood sugar are going to be cells of the pancreas and cells in the hypothalamus. The situation is very complicated because if the pancreas detects elevated blood sugar, before it releases insulin, it's going to communicate with the brain to see if the brain agrees with it. Okay, it's going to seek reinforcement that there's something wrong with blood sugar. So the pancreas is going to communicate to the brain. Cells in the hypothalamus are going to say, yeah, I'm also detecting the same thing. And then the cells in the brain are going to act as the control center to say, you know what, this really is out of whack. So I'm going to order you, the organ that responds to an imbalance in blood sugar, to do something about it. Okay. So these other parts of the body which take corrective action are going to be known as effectors. Okay. Number 3, let's define, oh, hold on. I missed a term I need to talk about here. So back up in control center, okay? Actually, a couple of things I need to back up here on. Okay, number 1, receptor. Okay, let's do a letter D here. Okay. When a receptor sends information into the control center, this is called afferent communication. Okay. Let's see. When a receptor sends information into the control center, this is called afferent communication. Again, folks, more vocab for you to wrestle with to sound sophisticated, educated, and fancy. Okay. It's just part of the language of the field. When the receptor sends information to the control center, when the pancreas sends information to the brain, when the pancreas sends information into the nervous system, that's going to be called afferent communication. Now I'm going to contrast this down below with the control center. When the control center decides that homeostasis has been disturbed, it's going to send information to the effector. And when the control center sends information to the effector, that's known as efferent communication. And the control center sends information to the effector. This is called efferent communication. All right, so what makes this confusing and what you got to pay attention to when you're studying is that obviously afferent and efferent communication sound a lot alike, and those words only differ by a single letter, okay? So just remember this. Okay. Remember, afferent communication sends information into the control center. And stated another way, efferent communication sends information out of the control center. Okay, so if you think about it that way, it becomes a little bit more potentially easy to understand, easy to memorize. Afferent is into the control center, efferent with an E is out of the control center. Okay, any questions right now?
Is this the same as what the textbook said about like input and output? So input would be afferent communication and output would be efferent communication?
As long as input and output are in reference to the control center, yes. Yes.
All right, thank you.
Obviously, input and output could mean different things if we change our perspective, right? So if the control center is sending information to an effector, the control center is delivering output. We're sending information out of the control center. But if I look at the information being received from the perspective of the effector, then the effector is receiving input. So you got to be very careful that you're defining what your viewing perspective is. Okay. All right. Any other questions? Okay. Let's define effector for you. Okay, effector. Effector is a body structure, typically an organ other than the brain, but it can actually be some subanatomy of the brain. Body structure, typically an organ other than the brain, okay, that receives information from the control center and takes corrective action to restore homeostasis. Okay, so our example here, okay, the cells of the hypothalamus of the brain said, yeah, there really is a problem with blood sugar. I'll just take the easy example here. Blood sugar is too high, so the nervous system is going to order the pancreas to release insulin, okay? Okay. The pancreas releases insulin in response to instructions from the brain. Okay. All right. So, you know, we started out with some nice examples of homeostasis. We talked a lot about blood sugar, and all we're doing now is putting some fancy words on things. Feedback systems, receptors, control centers, effectors. Okay. All right. Gang, do you have any questions right now? All right. So I tell you what I want to do. I want to go through one more example of a feedback system where rather than having me talk a lot, I want you guys to provide answers to my questions. So I'm going to be randomly choosing students from the room here. I'm going to be asking different questions. Don't get nervous if I call on you. It's okay to be wrong. You don't have to be perfect, right? I'm not perfect. People are not perfect. I just want you to try. Yes, a question, a comment on the chat window. Yes, these notes are going to be posted on Wiley by the end of today. Okay, they will be there. I would, unless I forget, they will always be posted on Wiley. Okay. Okay. So let's go through a hypothetical example. Hypothetical example of homeostasis and feedback systems. Okay, so here's what I'm going to do. I'm going to define this hypothetical scenario. Okay, let's assume the following statements are true. Okay. Number one, so let's say you are inside your house on a winter day. Number two, let's assume that you notice by looking out the window that the post office guy just delivered your mail. Okay. You see through your window that the post office just delivered your mail. Okay. Number three, you decide, hey, there's the mail. I want to go get it. So you walk outside to get your mail without putting a jacket on. Okay. Number four. As you're outside, you're getting cold, so you begin to shiver. Okay, so trying to give you enough information without giving you too much information so I can ask you some questions. Okay. All right. So let's see what we can do here. And I'm just going to be kind of random with how I approach this. Okay. So again, when I call on you, please unmute your microphone and say hi. And then just do your best to answer the question. Okay, so here's the first question. In our hypothetical example here, what body condition is being monitored? Okay, so I'm going to give, obviously I want everybody to think about the questions, but I just want one person to attempt to answer it. Okay, so I'm going to start with Layla Beck. Good morning, Layla. Please say hi to us.
Hi.
Okay, Layla, glad you're with us today. So Layla, what would you say about our hypothetical scenario? What body condition is being monitored?
I'm not 100% sure, but I kind of want to say temperature.
Okay, I love it. You happen to be right, and even if you weren't right, don't be afraid to answer. I'm not going to snap at you for being wrong. I might get a little excitable, but really, this is a very safe place. Okay, so yes, you're absolutely right. Body temperature is being measured. Okay. All right. Thanks, Layla. Okay. So second question. What stimulus created the change in body temperature? Okay, what stimulus created the change in body temperature? Let's go with, let's see here. I'm just looking around at my list here. Okay. Okay. Jordan Kyle. Good morning, Jordan. Please say hi to us.
Hi.
Jordan, what would you say with our scenario here is the stimulus?
The winter day.
Okay, can you be a little bit more precise?
It being cold outside.
Okay, I'll run with that. So doesn't it seem sensible that you just exposed your body to cold air? That's the stimulus, cold air. Okay. All right. Question 3. Good job. Good job, Jordan. Okay, question 3. What is the receptor in our example here? What is the receptor? Okay, so I'm going to ask this one to Kelly Chodkowski. Kelly, please say hi to us.
Hi.
Good morning, Kelly. Kelly, what would you say is the receptor in our example?
I guess that the receptor would be like your general like skin, body temperature, like that's the I'm mixing up the receptor. I'm sorry.
Okay, you want to think about it a little more, or
Um,
so you're telling me it has something to do with your skin? Is that what you're telling me?
yeah.
You know, that's not a bad starting point. It just would be a matter of, do you know what the anatomy of your skin is, so you can make a more precise answer to this? So, would you like to say anything else?
I don't know.
Okay, Kelly, you're doing okay. We're just getting started here, so thank you for attempting it. Folks, the receptor in our example are going to be nerve endings. Nerve endings embedded in your skin, right? Nerve endings embedded in your skin. Okay. There are going to be, so when I say a nerve ending, what the heck does that really mean? I'm talking about a nerve cell, a neuron, that has receptors on the surface of the plasma membrane that physically change their shape in response to changes in temperature. Okay, that's what I really mean at the molecular level. Okay, so at the molecular level, we are talking about a protein stuck in the surface of the plasma membrane of a neuron, where the protein changes shape in response to changes in temperature. Let me say that one more time. We're talking about a protein stuck in the surface of the plasma membrane of a neuron that's embedded in your skin. Where the protein changes shape in response to changes in temperature. If the idea of a protein changing shape in response to a change in temperature is odd to you, think about something you know in everyday life. Do you have, in your home or in any home you've ever been in your entire life, have you ever experienced a door that was harder or easier to open when it was winter versus summer? Winter versus summer. changes in temperature. If you have a wood door, right, typically in the summertime, heat causes expansion of the wood, so it might get harder to open and close. In the wintertime, air temperature is colder, so the wood itself of the door will compress a little bit and the door becomes easier to open. Okay, that's what I'm talking about. And this is reflecting an idea that's going to come about again in chapter 2 when we talk about chemistry, the fact that changes in temperature are one thing that will change the shape of a molecule. Okay, so it's all tied together and it's complicated, but it's doable. So the receptor in our example are nerve endings embedded in the skin. Question 4. Question 4, what is the control center? What is the control center? So let's just give this one quickly to, let's see, Brianna Weiser. Brianna, say hi to us.
Hi.
Hi, Brianna. So Brianna, what would you say is the control center?
Probably the brain.
Okay, nicely done. This is one of those questions where it's almost impossible to be wrong if you say the brain. And for our level of instruction at this moment of how we're thinking about content, brain is a perfect answer. Nicely done. There are going to be cells, the hypothalamus is one of those. The hypothalamus is a subsection of the brain that contains a lot of cells involved in maintaining information about various homeostatic processes in the body. So it's going to be the brain, specifically cells in the hypothalamus, okay? All right. Question 5. Question 5. Was the communication between nerve endings in the skin to the brain afferent or efferent? Okay. Kelly Lee. Kelly, please say hi to us.
Hey.
Hey, Kelly. Kelly, what would you say? Is this communication afferent or efferent?
Is it afferent?
Okay, nicely done, right? If we're moving information into the control center, it's afferent. Pretty simple stuff. You guys are catching on. Question 6. In our example, what is our effector? What is our effector? So I'm going to ask this of, well, let's see here. I mean, I'm just going to assume that certain folks are on. Let's ask this of Marie Huslin. Marie, are you with us today?
Hi.
Hi, Marie. Marie, did I mispronounce your last name?
No, it's Huslin.
Huslin. I'm going to have trouble with that.
That's okay.
Marie, what is our effector here?
Would it be the skin again?
Okay, it would not. You want to reconsider? So Marie, let's go up here and read our some assumed statements here. You're inside your house on a winter day. You see the post office delivering your mail. Number 3, you walk outside without a jacket. Number 4, you go out, as you're outside, you get cold, so you begin to shiver. So Marie, what anatomy of your body is most responsible for shivering?
Is it your brain?
No, not
Sure.
in this, not in the language of what we're talking about right now. The brain is more of the control center, and I'm asking you about the effector. What piece of anatomy of your body is most responsible for creating heat? Marie, what anatomy of your body is most responsible for the physicality of the shivering process?
I can't think of it.
Okay, you tried. Thank you, Marie.
Sorry.
So it's okay. There's no need to apologize. Okay. So for everyone else, I think maybe you might be thinking of your skeletal muscle, right? Your skeletal muscle is the effector, right? Because the skeletal muscle is going to respond to information from the control center, from the brain, where the brain tells your skeletal muscle, contract and relax repeatedly. Contract and relax repeatedly. That's what shivering is. That will use ATP, biochemical energy, to contract the muscle tissue. And when you break down ATP, just like when you burn gasoline in your car, there's a little bit of waste heat, right? The hood of your car gets warm because you're burning gasoline. Your body will get warmer because you're burning, quote unquote, gasoline inside the body. Marie, does that make sense?
Yes, it does.
Okay. All right. All right, let's see. So I kind of answered my next question. So I'll pose the question and I'll answer it. Why did the skeletal muscle shiver? The answer is because the control center, the brain, ordered it to. They didn't have a choice, right? Nerve impulses went down the spinal cord and into the skeletal muscle tissue, and the brain said, I want you to contract. And so they contract. Okay. Question 8. Okay. Was communication between the brain and skeletal muscle tissue afferent or efferent? Okay, so let's do Jada Davis. Jada, say hi to us, please.
Hi.
Hi, Jada. What do you say for this question? Is the communication between the brain and skeletal muscle afferent or efferent?
Is it the efferent?
With the E, nicely done. Thank you, Jada. Question 9. Okay. All right. Question 9 is this. Is our example here a case of positive or negative feedback? Okay, so we haven't talked about this yet. This is a little new wrinkle here. Okay. And so I'm going to pick someone that I think needs a challenge. So let's try Kendall King. Kendall, can you please say hi to us?
Hello.
Hi, Kendall. Kendall, in terms of our current situation we're talking about, would you say the response of the body is an example of positive or negative feedback? Please repeat yourself.
I'm sorry, I'm trying to think here. I would probably say negative feedback because the situation wasn't actually corrected. Our body did start producing more heat, but that heat would be let off and we wouldn't get that heat in if we're still out in the cold without a jacket. That's just an assumption. I'm sorry.
Okay, so you said a lot of things there in your answer. And you know what? I like the statement that it's negative feedback, but I'm not entirely comfortable with how you tried to explain it afterwards. So, you know, you did a good job. You gave me the right answer. The reason was a little bit fuzzy, but you did a good job, Kendall. So thank you. Okay.
Okay, thank you.
So this is an example of negative feedback. Okay, so how am I going to know whether it's negative or positive? Here's how you know, folks. Okay. Negative feedback works to reverse the effects of the stimulus. Negative feedback works to reverse the effects of the stimulus. Okay, think about it. The stimulus was the cold air, which is making our body cold. So what did your body do? It started to shiver, to create heat, to warm it up, to counteract the effect of the cold, to reverse the effect of the cold, to reverse the effect of the cold air. Therefore, the response of the body in this circumstance was a negative feedback response because the body attempted to reverse the effects of the stimulus. Your textbook tells you, and it is true, that most feedback systems in the body are of a negative example. Typically, if you have something out of balance, out of homeostasis, your body will try to counteract it, will try to reverse it, right? If your blood pH is too low, your body will take action to elevate the blood pH. So that would be a negative feedback circuit, a negative feedback scenario. There are, however, examples of positive feedback. Okay, positive feedback works to enhance, or we might say amplify, the effects of the stimulus. Okay, and while it is true that positive feedbacks are less common, they're not completely uncommon. So let me give you a couple quick examples of positive feedback. Okay. Okay, so examples of positive feedback would be, I think one that your book immediately gives you is uterine contraction during childbirth. Okay, that's one example. Okay, so typically uterine contraction during childbirth is stimulated by a hormone called oxytocin. Some of you gals that have been through the birthing process may have had a chemical cousin of oxytocin administered by your doctor, your physician, called Pitocin to help stimulate the uterine to contract, to push that kid out quicker, right? As that contraction process begins, it's a self-amplifying effect because we really want to continue to powerfully contract the uterus to get the child out of the body. That's an example of positive feedback. Blood clotting. Blood clotting and the formation of the blood clots. When you activate platelets to clot the blood, the activation of platelets will then stimulate additional activation of more platelets. So it's a self-amplifying effect here. Let me give you one more that it's not going to mean anything to you in terms of detail because we haven't covered it yet. But there's this thing that I'm kind of referring to about communication between the brain and other areas of the body. Well, that communication is electrical in nature. It's something called a nerve impulse. And a nerve impulse, the propagation of a nerve impulse, is a very classic and important example of positive feedback. Okay, folks, question in the chat window. Are you with me? Are you okay? Is there anything else I can try to clarify for at this time? Okay. All right. Please let me know if you need help. Question 10. All right. So, considering our example, if you were standing outside on a cold day, what action could you intentionally do to create positive feedback to the stimulus? Let me see here. Let's see. Courtney Cook. Good morning, Courtney. Can you please say hi to us?
Good morning.
Good morning, Courtney. So think about this question. If you're already cold because you're outside on a cold day without a jacket, what could you do to create positive feedback?
Run around.
I'm sorry, I may have missed what you said there. Are you just thinking?
I said run around.
Run around. Okay. So, okay, Courtney, stay with me, and everybody else, please stay with me here. Okay, so question in the chat window. So in relation to question 10, Courtney said run around. Hold on. Here we go. Do you agree with Courtney that running around provides positive feedback? Okay, okay. I'm so glad I asked this question. Okay, folks, remember, it's my job to lovingly give you a kick in the pants every once in a while. That is not correct, okay? Think about it. If you run around, you're going to be contracting muscles to run around, which means you're burning energy to create heat, which is going to warm you up, which is a negative feedback. So running around will most definitely not be a positive feedback scenario. It will be a negative feedback scenario. Okay, so let's just put some language on this. Okay. So my first point here, and don't worry about this, Courtney, because many students fall into this kind of trap, and don't feel bad about it, is that you have this psychological need, this emotional need as a person to say, well, if I'm cold, I need to run around to warm up, and warming up would be positive because I don't want to be cold. I don't like to be cold. This is not a psychological thing. This is a biochemical principle. Your response to the question has got to be in response to what it means to have positive and negative feedback. And that always references the stimulus. If the stimulus is the cold air and the cold air is making you cold, then the positive feedback that you want to do should enhance the cold air. So Courtney, take a second stab at this. What could you do to enhance the stimulus?
I'm not sure exactly.
Well, think about it. It's a winter day. So what might be outside on a winter day?
Could be snow.
Okay, so what could you do?
Sit in it.
Okay, beautiful answer. You could sit in the snow. Okay, you could sit in the snow. It would be a little crazy, but that would make you colder, wouldn't it? Okay, okay, good. All right. Good, good. Nicely done, Courtney. Okay, so just be careful there about that positive and negative feedback. Resist the urge to take a psychological or emotional approach to these kinds of things. When you answer positive, negative feedback questions, you got to think about, well, what is the stimulus doing? And if I want positive feedback, I got to enhance the effect of the stimulus. If I want negative feedback, I got to reverse the effect of the stimulus. That's the part that I see students getting tripped up on most commonly. Okay. All right, folks. I think I'm going to let it rest. So do you have any questions right now about homeostasis, feedback systems, the terminology, receptor, control system, effector, positive, negative feedback, anything I can help you with? Okay, so folks, we are done with chapter 1, which I'm very happy with. I won't spend any more time lecturing on chapter 1. I will start lecturing on chapter 2 after Labor Day. By the way, let me just wish you a nice Labor Day weekend. Hope you have plans to have some fun. So, okay, so I'm going to momentarily stop sharing my screen. Okay, we're done with lecture now. We're currently in our lab session, and so we're going to get started with lab content. We have lab typically most days from 9:30 to 12:10. Generally speaking, I will not record lab, but I've continued to record right now because I was lecturing. And I will at least record part of lab here because I'm going over brand new first time things that maybe you would want to be able to review in a recording in the future, okay? So, and also let me say this, lab time will be more of an individual effort on your part, and I won't necessarily keep us here the whole time every class. And, so, I will keep us as long as I feel we're being productive. But to be honest with you, a lot of what you have to do requires private study time. So hold on a sec. I do see a comment in the chat window. Are we responsible for content from section 1.7? I'm assuming you're asking in the textbook. Yeah, okay. All right. Yeah, okay. So, so are you responsible for the content of section 1.7? And my answer to that is I want you to read 1.7. I want you to read it. That being said, I don't intend to badger you with a lot of complicated questions on medical imaging. If you need to be an expert on medical imaging technology, you will get that in more advanced, I won't say more advanced, but more specialized courses in your future medical career. So reading medical imaging is definitely a worthwhile thing, but no, it will not be an emphasis of my future exams. Another question, do we have any homework for lecture? Courtney, I do not explicitly assign homework for lecture. I do assign homework for lab. That's your lab homework that I'm going to continue to talk about now. You remember in Wiley Plus, in the lecture component of Wiley Plus, I do have some suggested activities for you to do. They're not required, but they are my recommendation that these are things you should look at. But no, there's not any homework for lecture that you've got to submit or turn in any way. Okay, anything else?
I can't remember if we went over this in class on Monday, but is there a place where there's like a calendar on all the chapters we're going to go over in class so I know what to read before lecture?
All of that's in your syllabus. All of that's in your syllabus. So let me show you a couple things just to remind everybody about things, and hopefully that'll help you out, Raquel. Okay. All right. So let me just share my screen with you all, and I'm going to minimize this document here, and I want to get rid of the cell size stuff. Okay. Okay. So hold on a second here. All right. So. Okay, so I'm going to bring up the lecture component of Wiley Plus. Okay, the lecture component of Wiley Plus. Here it is. Okay, so right now I'm in the lecture component of Wiley Plus, and this gets to one of the questions. Here are assignments in the lecture component of Wiley Plus that I'm recommending for each chapter. You don't have to do these. They will not be part of your grade, but they can help you evaluate whether you're understanding material properly or not. So these are recommended things. Now, Raquel, in terms of the assignments that are due, you would go to Files, you would go to your syllabus, and I encourage you to get your syllabus and download it to your computer, right? Download it to your computer, and if you have a printer, I even encourage you to print it out, because that document can answer many of your questions. Here's your reading schedule for the course, right? You can see that on Wednesday, the September 8th, I will be lecturing in chapter 2. So if you get a chance to read into chapter 2 over Labor Day weekend, that would be a good thing. Also, your assignments that are going to be due in the lab component of Wiley Plus are listed in the syllabus as well. Okay, so having the syllabus easily, fast, readily accessible is helpful to you. Okay. Also, I wanted to point out, once again, in the lecture portion of Wiley Plus, under pages, I created a link to lecture recordings, right? And so I already posted the opening day lecture. Let me just pop this open for you. Okay, so there's my. Okay, good morning, folks. We'll just continue. That's my ugly mug from Monday. Okay, so all these lecture recordings will be posted to YouTube, and I will provide the links in Wiley Plus. All you have to do is go there and press what you want. Okay. Any questions so far? Okay, so let me ask a question in the chat window. Have you successfully accessed your textbook through Wiley Plus? Do I have anybody that needs me to go over that again? Okay, I'm not hearing anybody that needs that, so I'll continue. Here's another question. Have you successfully accessed your lab manual through Wiley Plus? Does anybody need me to go through that? Yeah, there's a separate lab manual from the textbook. Does anybody need me to go through that? Okay, Noah, do you need me to go through that?
I think somebody had a question about how to download the book, just in case they don't have any Wi-Fi or if they're traveling.
Yes, I honestly don't quite remember. Once you have your book loaded up in the web browser, there should be a settings tab along the left-hand column that gives you the option of downloading it. Am I saying something that rings true to those that have downloaded it? Okay, so Jada's saying yes. Okay, so if you can bring up the textbook in the web browser, then along the left-hand side, as you manipulate the menu on the left-hand side, you should be able to find like a ratchet button, a settings button that gives you the option of downloading it. Okay. All right. So, okay. So this is the lecture part of Wiley Plus. Now let's go to the laboratory part of Wiley Plus. For the lab part of Wiley Plus, which I'm in now. Okay. Notice the assignments come up. Whoop. No, that's not true. That's not what I wanted to show you. Sorry, folks. Okay. All right, that's not what I wanted. Okay, let's do this again. Love technology. Okay, let's go to the lab. Here we go. Okay, so I'm now in the lab portion, and you can tell it's lab because the assignments always say lab exercise 1, blah, blah, blah. So this is the lab component. Right here are the lab exercises that are assigned to you and that are due next Wednesday. Okay, lab exercise 1 activities is due on September 8th, reviewing your knowledge, September 8th, using your knowledge, September 8th. Same thing for lab exercise 2 activities, reviewing your knowledge, using your knowledge. These are all due September 8th, which means, unfortunately, probably you'll have to do some work over Labor Day weekend because exercises 1 and 2 are due, meaning they must be completed and submitted by 9:30 a.m. next Wednesday. If you miss that deadline, the computer will lock you out, okay? So you really do need to submit by 9:30 on September 8th. The lab homework is required for the course. You must do it. And the reason you must do it is because I need you swimming through the content and building your knowledge. The lab homework, as difficult and lengthy as it might seem at times, is really to build you up so you can be a really capable future rad tech, dental hygienist, nurse, whatever it might be, whatever field you're going into. We need to build you up. Okay, so those are the things that you'll do. And these will be graded. These things you do and how well you do on them will count towards your final course grade. Any questions so far? Okay, all right. So what we want to do now is answer the question, okay, so I now know what my lab homework is, and I know when it's due because I can tell by the information on Wally Plus. And I also can read my syllabus and get an idea of what it says there, so I can do either thing, okay? What exactly do I need to do for lab? What are lab exams going to be? Well, for lab exams, you're going to be responsible for knowing anatomy. So what anatomy do I need to know for the first lab exam? And that is going to be the anatomy of the skeleton. So if you are in Wally Plus, so Rachel Hoffman asked me, are these timed homeworks? They're not timed when you're doing the homework, Rachel. What I mean is, you know, if you want to take 15 minutes to answer one question, that's okay. They are timed in the sense that you must submit the work by next Wednesday, September 8th, but the homework itself is not timed. I'm not putting time pressure on you. Okay. Any other questions? Okay. All right. So if you are in Wally Plus, in the laboratory portion of Wally Plus, go to Files, and you will see different word banks. Bones for lab exam 1, muscles for lab exam 2, nervous system for lab exam 3. And this tells you how the course is going to proceed here in lab. The first 1/3 of your lab curriculum is learning the bones of the skeleton. The second third of your lab curriculum is learning major skeletal muscles of the body and honestly some of the tissue. So that's a little bit of histology, which speaks to this thing here, histology slides. The final third of the semester will be understanding nervous system anatomy, brain anatomy, spinal cord anatomy. Okay, so don't worry about all these other things in the future. Just focus on what's necessary now so you don't get overwhelmed. So go ahead and click on Bones for Lab Exam 1 in the lab portion of Wally Plus under Files. Okay, these are the bones and different features of the bones that you are responsible for being capable of identifying. Okay, there are 5 pages. There's a lot of content here. There's a lot of content here, and you will have to spend significant practice time learning this anatomy. Some people memorize things faster than others. That's just the reality that we're all different people, but you're going to have to put the practice time in that you need to put in to learn this. Okay, so today, excuse me while I get my proper syllabus out here. Today is September 1st, and I'm introducing you to this content. You have your first lab exam on September 29th. So 4 weeks from today, you will be testing on this anatomy. So in 4 weeks, basically 1 month, you must learn this anatomy sufficiently well that if I show you a picture of a bone, you can tell me what the bone is. If I show you a picture of a bone in a section of the bone, like the olecranon, you can tell me what that is. For a lab exam, you do not have to tell me the function. I'm not going to ask the function. What you have to tell me is the identity of the object I'm asking for. Okay, so I very strongly, again, I strongly urge you to open up the file on Invisible Body and download it to your personal computer. Create a folder on your computer that's Biology 121 and keep important information like this on your personal computer, keep it organized. If you have the luxury, I made a mistake last semester when I assumed that a student had a printer and I realized the student didn't have a printer. I was like, oh my God. Okay. I know maybe you don't have a printer. I'm hoping you do. If you have a printer, please print out this and make a hard copy for yourself. It will make life easier on you. Because if you've got to have this up here and then have another window up on your screen with visible body and your textbook, all of a sudden you don't have enough space on your computer screen anymore and it gets really cumbersome. So download and print out what you can. It'll make life easier. Okay, question coming? Yes.
So like, is there on visible body? I haven't really logged on yet, but you can print out like diagrams of a skeleton and label it, like any of that?
Okay, I'm going to show you how to use visible body today. Visible body is not terribly good at printing things, but it's basically very good at everything else. Okay, so I will get you started today with it. I'm not going to show you everything because it can be overwhelming, but you will learn this through the process of me showing you features, you pushing buttons and seeing what happens, because you can't break it. Okay, you can't break it, so don't worry about that. Okay, so let me just ask everyone through the chat window. Can you bring up the Bone word bank on your computer? Does anybody need help with simply getting to this stage?
Could you just go over it one more time how you got there? I was trying to figure it out and I didn't have my Zoom up to watch your part.
Okay, all right, so let me close this out. And so what I'm going to do is I'm back at the laboratory component of Wiley Plus, the lab component of Wiley Plus. I'm going to go on the left side to the Files tab. And that's where you find your bone list. Bones for Lab Exam 1, you click on that. Does that help?
Yep, thank you so much.
You're welcome. Okay, so now that we've got everybody with their bone list, you know, I have a physical copy printed out for myself because I just like to work with the physical copy. So I'm going to close my bone list on the computer and simply talk to you about the bone list that I'm holding in my hand on a piece of paper. Okay, but you can keep your bone list up on your computer if you prefer to do that, or if you prefer to download and print, I encourage you to do that. Okay, so now what we need to do is get visible body started. So back on Wiley Plus, what I want you to do is go to the Pages tab on the left. Go to the Pages tab on the left. Go to the Zoom and visible body links for class. So remember that for you to access visible body, you must be logged into your MyHack account. So if you're not logged into MyHack now, please do it. I'm going to give you a few seconds to do that. Please log into MyHack. Okay, so as you are doing that, as you are logging into MyHack, obviously the next thing you do is click on visible body. So I'll just do that. And you notice it brings up visible body. It brings up this web page. It says Human Anatomy Atlas 2021. And in the middle, it says Launch Human Anatomy Atlas under a yellow ellipse of sorts. So when you're ready, you've logged into Wiley Plus, you've logged into MyHack, and you click on this link, go ahead and load it up. And so I'm just going to stop right now and ask you through the chat window. Okay. There we go. Is visible body loaded on your computer? Okay. All right, so I'm seeing a not yet from Sharia. Okay, so most of you have it, but let me say just a couple things here. What I have encountered over numerous years now is that sometimes there are a handful of students that do struggle to get visible body loaded on their computer. And the reason can be a number of things. Number one, if visible body is not loading properly on your computer, it could be because you're using the wrong web browser. Once again, Google Chrome is the preferred web browser for this software. Secondly, if you sit there and all you do is see something kind of spinning and nothing's loading, that often indicates a problem with your internet connection. It could be a weak internet signal. You could have a problem with your internet bandwidth. If you've got too many people at home trying to surf the web at the same time, that can be a problem. Okay, so please let me know if you need extra help just getting visible body to run. Okay, so anybody want to speak up about a problem right now?
Yeah, where did you find the visible body at?
Where did I find visible body at? Okay, hold on a sec. So visible body, what I did was I logged into Wiley Plus in the lab portion of Wiley Plus, and I went to the pages link on the left side. There's a link there for visible body. Does that help? Okay, anybody experiencing just a traumatic experience right now? Nothing's working.
Me, I am. I'm trying to download Chrome so that way I can see if it resolved my issue.
Okay, I tell you what, for the sake of moving others through, I want to continue, but I can come back and give, some private information, some personal time for those that might be struggling, okay? All right.
Okay, thank you.
So do the best you can, and I promise I won't leave you out there. I will come back around to help. Okay, so for those that successfully got visible body loaded up on their web browser, first know that this is really very powerful software. I used to require my students to purchase this, but recently I was able to convince the omnipotent supervisors at Hack to spend money on this thing. And so Hack purchased some licenses that you as a student can now access, and I don't have to get you to buy it yourself anymore. So that's a nice thing. This is award-winning software. It's used in medical schools around the world, and you have access to it now. And this is how we're going to learn anatomy. So first off, if your opening screen looks like my opening screen, let's just take a quick look at a few things. Number one, starts out with the Views tab, the Views icon on the left highlighted blue. If we go to Media, go to Media, and you'll notice that there's just different animations. For example, I'll just click on one.
Cells are the basic living units of tissue. The human body is composed of...
Okay, so there's actually some videos here that are not only relevant for anatomy, but also physiology. And you might want to remember that when you're studying some physiology, that there are some animations in your visible body product that you could potentially access and might be helpful to you. Okay, so there's media. There's also quizzes. And by quizzes, I mean that the people that manufactured the software have set up some pre-made quizzes to challenge your knowledge of anatomy. Their quizzes won't necessarily be my quizzes. Their quizzes are not going to be my exam, but they can still be helpful to you as you're learning things to see if you can answer a question, okay? So, eye expression, okay? It says here, select the right or left levator palpebris superioris. Well, I'm just going to randomly pick something and see what happens. Okay, submit it. Oh, that was incorrect. I selected the occipitofrontalis muscle. Okay, so there are pre-made quizzes here on certain pieces of anatomy that you can do when you want to. I'm going to go back to the main menu here. There's a My Library tab. I won't talk to you about that just today, but there is at times the ability to create your own sort of virtual digital flashcards and things like that and save it. Okay, we'll talk about that later. Views. Let's go back to views. Okay. So under views, we have a subcategory of menu items. Let's go to regions, right? So there are region views. There are system views, which honestly is going to be your major starting point for most work that you do. Gross anatomy lab. There are cross sections. There's microanatomy. Microanatomy will be actually very helpful when we're learning the nervous system at the end of the semester. And there are muscle actions. By muscle actions, I mean when the muscle contracts or relaxes, how does the body respond? How does the skeleton move? And these are actually very nice. If I click on this, right, you can see that it's going to show you the motion of the skeleton when the actual muscles are contracting and relaxing. Okay, so that can be very helpful when you're learning the muscles and what they do, how they manipulate the anatomy of the body. Okay, so all good stuff, and my point here is that you have a lot of sophisticated functionality at your fingertips. It can be overwhelming at first. If you ever get flummoxed with something on the screen and you just want to start over, in the lower left is the menu button. You can click on that and bring you right back to your main menu. Okay, so I'm back to my main menu and I'm going to go back to systems. So right now I have view systems located. Okay, so before I go any further, do you have any questions? Okay, let's show you how to use this software. Okay, we're just going to get started here. Let's go to view number 2 for the skull. So click on the skull. Okay. Now there's a couple ways you can work this. And I'll show you 2 alternative ways to do the same thing. Okay, if you're using a standard computer with a mouse, if you hover your cursor over the bone, over the skull in this case, and you press and hold the left mouse button, you can rotate the skull. You can rotate the skull. Okay, that's with the left mouse button. If for some reason you're working on a device where you don't really have a mouse, like a tablet or something or whatever, you could use this joystick down here and kind of press on the center knob of the joystick and rotate the skull around that way. So just see if you can figure out a way to rotate the skull with what technology you're using. Secondly, if I don't want to rotate the skull, but I physically want to move the entire object, right, left, up, down, what I'm doing is putting the cursor on the skull and pressing and holding the right mouse button, And if you don't really have a right mouse button, well, you can click these arrows on the joystick, left, right, up, down, right? So either way is fine. You do what is most comfortable for you. And finally, if you want to shrink or magnify the object, on my computer at home, my mouse has a scroll wheel. So what I can do is use the scroll wheel. If I scroll down, I shrink the skull. If I scroll up, I magnify or enlarge the skull. If you don't have a scroll wheel on your device, you can go down to this slider window below the joystick and you can shrink it down or magnify it that way. So please try those controls out on your device a little bit at this time. And I'm just going to ask you in the chat window. Can you rotate, move, and magnify, shrink the skull? Okay, fantastic. You know what, gang? You're 90% of the way through the battle here, because that's basic functionality for the software that you're literally going to perform those actions thousands of times this semester as you plunder through the anatomy lists. Okay, so let's get started with something. Okay, so any questions? All right, so obviously you're free to play with the software on your own time and learn things. I'm just showing you some basics here. If you have the skull open on your front screen like I do, press the frontal, put your cursor on the frontal bone and click the left mouse button. There you go. When you click the left mouse button, what it does is tell the software, I want you to highlight that bone or structure, in this case, the frontal bone, and it brings up a selective structures window to the right of the object. There's a lot of functionality in here that you can play with. I want to point out a couple things for you. Number one, there's this icon up here that looks like a textbook. If you move your cursor over it and open it up, then it will give you some textual description of the object that you've selected and tell you about it and related structures that it might be connected to. So this can be helpful when you just want to learn a little bit about the object that you're clicking on, okay? Obviously, the other place you can go for information and which you should not forget about is your textbook. Your textbook is magnificent, so please use it. Okay, so with that stated, the textbook will give you information about the object. Let's click the textbook again to hide it, to hide that information. Okay, so what I want to do next is, with the frontal bone highlighted blue, I want to remove it from the skull. So go over to the hide button and click on hide. Here I go, 1, 2, 3, hide. And the software will remove the frontal bone. Okay, so at this point, you have a really nice way of rotating the skull towards yourself so you can look down into the cranial cavity and see the space that would normally be occupied first by your brain and below the foramen magnum by your spinal cord. Okay, so we're looking into your skull. Okay, all right, so I have a question for you in the chat window because I want to be sure you're accessing information correctly. So hold on a second. In the chat window, here we go. Oh, that didn't go to everybody. It was supposed to. I got to do that again. Hold on a sec. Here we go. What is the name of the bone on the first page, lower left corner of your word bank? What is the name of that bone? Okay, so I'm seeing some answers that are incorrect. Okay, the answers I'm seeing so far are incorrect. The word bank that I'm talking about is the list of bones. Where is this word bank? That word bank is in Wiley Plus. Do I need to go over that again? Okay. So in Wiley Plus, the bone word bank that I showed you how to access, that's the word bank I'm talking about. Okay. So Brittany, you're saying it takes a minute to pull it up. That's why I would encourage you to download it and if possible, print it. Okay, it will make life easier if you can do that.
Definitely
Okay.
will do that.
All right. Okay, so I'm just going to run with Shay's comment, the sphenoid bone. That's exactly right. Do you see in the word bank the term sphenoid bone? Okay, you see Shay's response, my response. Folks, the bones are listed in bold-faced terms, right? You notice that it's bold font. So you have to be capable of identifying all the bones, which are bold-faced font, and all of the terms underneath each bone, all the terms that are normal font, that is not bold, those are what are referred to as landmarks or features or substructures of the bone. You're responsible for knowing those as well. Okay, so we're going to start that today. Any questions before I continue? Okay, so let's find the sphenoid bone. And well, I'm just going to show you where it is. The sphenoid bone is this bone right here that has on its top surface, it kind of looks like a butterfly. It kind of looks like a butterfly or a bat, depending if you like Halloween or something like that. Okay, but this is the sphenoid bone. Go ahead and highlight the sphenoid bone. Okay. With the sphenoid bone highlighted, I want to learn all of the substructures on that word list that I need to know, such as sinuses, sella turcica, greater wings, lesser wings. I need to know that. So what you do is you highlight the bone and you go up to the selected structure and you click on the details button that's multicolored. Click on that. Oh my goodness, look what we have here. We now have a multicolored, rainbow colored sphenoid bone. And each of the colored segments is going to be some kind of landmark or substructure of the bone, some of which you are responsible for identifying. Okay. Any major problems at the moment? Please speak up. Please speak up. Okay. Now, what I want to show you, wait. Okay. Okay, Kendall, thank you. What I want to show you is, you can pick any of these colors, okay, right? Greater wings, lesser wings. The software is going to tell you what this stuff is. Here's the deal. Let's do this. Let's take a look at the lateral pterygoid process. Say, so pick this red guy at sort of the bottom right of the bone. Pick that guy. Now you might say to yourself, I don't even know how to pronounce that word. Well, we got a solution for that. Click the speaker icon on your software. Go ahead, click that.
Lateral pterygoid plates. Lateral pterygoid plates.
Okay, my point is that all these difficult to read, difficult to pronounce, difficult to understand words, the software is going to support you in knowing what it looks like, what it is, and how to say it. Okay, and so you can work through here as best you want. This is one of my favorite sections of the sphenoid bone to highlight. This is the sella turcica. Okay, if we read up here, the sella turcica is a saddle-shaped depression that contains the pituitary gland and its deepest part of the hypophyseal fossa. What this is telling you, folks, is that if your brain was part of this image, your pituitary gland would be nestled down in this crevice, in this ridge called the sella turcica. Okay? So, long story short, it is your job to learn all the bones and their landmarks and their substructures that are on the physical list on Wiley Plus that I've showed you how to access, and I'm encouraging you to print it out for yourself. Okay? You are responsible for knowing them and being, when I say know them, I mean being capable of identifying them. I will not ask you the function. I will not ask function. I mean, function is important, but you're already going to be overloaded with identification. You're going to be overwhelmed with identification. So we start with identification. I'm going to ask you to identify any of the terms on this list, okay? Do you have any questions right now? All right, so let me make some other comments here, and then we're going to do a little activity to kind of break the ice here. Visible Body is a fantastic piece of software, and it gets updated basically on a yearly basis. So a lot of times there's some new functionality that even I'm not aware of because they're constantly updating it. There are some pieces of anatomy listed in your word bank that the software won't do a very good job of. For example, on the front page of your word bank and the top middle column, it says cranial fontanelles of the infant skull. Well, as far as I know, the software does not include the infant skull. So you ask yourself, well, how can I learn the infant skull when it's not in Visible Body? And my point in bringing this up is anything you really cannot find in Visible Body, you need to go to your textbook for it. You need to go to your textbook. So let's have a very friendly competition here. I'm going to put this into the chat window. Okay. Hold on a sec. Okay, so here we go. Who can be the first person to list the page number in your textbook, not the lab manual, the textbook, where a picture of the cranial skull and fontanelles is found, of the infant cranial skull and fontanelles is found? What's the page number, folks? Let's have a friendly competition. Who can be the first to get it? What page do I find a picture of the infant skull and cranial fontanelles? Okay, so I see one page number listed up here. I'm going to check it. Okay, page 198, I don't think is correct. It doesn't seem to be correct in my physical book. Okay, so I see that one of you reported chapter 7. Good, that is chapter 7. And so I'm seeing some of you starting to report page number 222. That's correct. Folks, everybody, please go to page 222 in your textbook. Go to page 222 in your textbook. And just have a look at that, okay? Take a look at figure 714, fontanelles at birth. Okay. All right. All right. Excellent. So, you know, I could ask any of you. So let me ask Morgan Bruni. Morgan, please unmute your microphone and tell us how you found this.
I just searched on, like in the textbook, you can search for words. So I just searched infant skull.
Okay. Good. That's what I was hoping you would say. Okay. So remember, folks, the benefit of having a digital electronic book is that you can search its contents pretty rapidly. So you could type in infant skull or fontanelle or something like that, and there's a good chance the book's going to locate it for you. Okay. All right. So any questions at the moment? Okay. All right. I know we're getting a little long in the morning, getting a little tired. I'm starting to feel it myself, but I want to get you started in one more thing so that we really break the ice here with this software. Okay, so what I'm going to do next, and for those of you that might be having some technical issues, I promise I'm going to help you. But for everybody else that seems to be okay with the technology right now, here's what I want to do. I'm going to go through the word bank, and I'm going to assign every person in class at this time a bone or maybe a couple bones, and I'm going to ask you to, in the next 20 to 30 minutes, become the class expert on that bone using visible body and your textbook as your references. This doesn't have to be perfect. I don't expect you to be perfect, but I want you to start the process of learning a bone. And then what we will do after 20 or 30 minutes of private study time is we will go into breakout sessions, breakout rooms, where each of you attempts to share your computer screen and explain the bone that you've been studying for the past 20 to 30 minutes. Okay? All right, so bear with me here. I'm going to call out names and assign you a bone. I would ask that after I tell you what bone you're responsible for, you will please give me a yes or no in the chat window that you heard me correctly. Okay. All right, so here we go. Okay, so Sharia, I'm going to ask you to peek. I'm going to ask you to learn the occipital bone, the occipital bone. Is that okay, Sharia?
Yes, it is.
Thank you. Okay.
You're welcome.
Rachel Hoffman, Rachel, I'm going to ask you to learn the temporal bone, the temporal bone.
Sounds good. Thank you.
Okay, great. Courtney Cook, Courtney, I'm going to ask you to learn the sphenoid bone.
Okay.
Courtney, let me tell you, as you look at your word bank, there are three terms to the right of the sphenoid bone: the foramen rotundum, foramen ovale, foramen spinosum. They are part of the sphenoid bone, so please include those in your study time.
Okay.
All right. Let's see here. Kelly Chodkowski. Kelly, please learn the ethmoid bone. Kelly, please learn the ethmoid bone.
Okay.
Thank you. All right. Hold on a second. Okay, Jordan Kyle. Jordan, I'm going to ask you to learn the maxilla bone, the lacrimal bone, and the zygomatic bone. So once again, Jordan, maxilla bone, lacrimal bone, and zygomatic bone.
Okay, thank you.
All right, Jordan. Okay. Let's see, Brianna Weiser. Okay, hold on a sec. I gotta answer a question in the chat window. Okay, so Courtney, you're asking about the sphenoid bone. I'm simply pointing out, Courtney, that on your word bank, in the middle column at the bottom, there are three terms: foramen rotundum, foramen ovale, foramen spinosum. Those go with the sphenoid.
Okay, thank you.
Okay. All right, so let's continue here. All right, and let's see. So I think the last person I assigned was Jordan. So I will go next with Brianna Weiser. Brianna, please learn the vomer bone, the palatine bone, the nasal bone, and the hyoid bone. Brianna, I'm assigning vomer bone, palatine bone, nasal bone, and hyoid bone.
Thank you.
Thank you. Okay, Kelly. Kelly, I'm going to ask you to please learn the mandible. Please learn the mandible bone.
Okay, thank you.
Okay. Marie Huslin. Marie Huslin. Marie, I'm going to ask you to please learn the vertebra that says atlas. The vertebra that says atlas. And I'm also going to assign Marie, please learn the vertebra that says axis.
Okay.
Great. Okay, so Layla Beck. Layla Beck. Layla, I'm going to ask you to learn the thoracic vertebra. And Layla, I'm going to ask you to learn the lumbar vertebra.
Okay, thank you.
Thank you, Layla. Okay. So for those that are studying the vertebra, and this is a general comment to everybody, when you learn one vertebra, that's a very good foundation for learning the other vertebra because they're very similar and have common features. So it might seem like it's a lot, but when you learn one, it helps you with the others. Okay. Jada Davis. Jada, I'm going to ask you to please learn the pelvic girdle. Jada, is that okay?
Yeah, that's fine. Thank you.
Okay. All right. Morgan Bruni. Morgan, I'm going to ask you to learn the following. Please learn the sacrum. Please learn the clavicle. Please learn the sternum. Please learn the rib. So Morgan, you're assigned the sacrum, clavicle, sternum, and rib.
Okay, thank you.
Okay, thank you. All right, so Jordan Benson. Jordan, please learn the scapula. Is that okay, Jordan?
Yep, thank you.
Okay, great. Okay, Israel Cologne. Israel, please, on the next page of the word blank, Israel, please learn the humerus. Is that okay, Israel, for the humerus?
Yes.
Okay, great. Thank you. Scarla Collado, excuse me. Scarla, please learn the ulna and the radius. The ulna and the radius.
Okay.
Thank you. Okay, I'm just taking notes here. All right, Kendall King. Kendall, please learn the hand and wrist.
Okay.
Okay, now Kendall, the hand and wrist is a challenging one because there's a lot of tiny little bones called carpal bones in the wrist. My recommendation to you is that you definitely locate the pages in your textbook where that information is covered, and that will help you get started with the software.
Okay, thank you.
Okay. All right, so let me see here. Okay, so please forgive me, the software is a little crazy here. Okay, I see. I'm trying to figure out who I did not assign parts to yet. Karen, did I assign parts to you? Karen, do you have parts assigned to you?
Not yet.
Okay, Karen, please learn the femur. Karen, please learn the femur.
Okay.
And also, Karen, please learn the patella.
Patella. Got it.
Okay, great. Brittany Singer. Brittany.
Yep. Yep.
Brittany, please learn the tibia and the fibula. The tibia and fibula. Okay, thank you.
Thank you.
Okay, Shay. Shay, I'm going to assign you the foot and ankle. Is that okay, Shay?
Yep, I got it.
Great. Shay, the same advice for you as I had for your classmate about the wrist. If you look up foot and ankle in the textbook and it can help you get started with the software.
Okay.
Okay, so we're going to wrap around and do some other, there's enough students here that I can assign some objects twice. So Noah, I'm going to ask you that you also learn the pelvic girdle.
Okay.
Great, thank you. Pelvic girdle is an interesting one, kind of quirky, a little challenging, but I know you can do it. Let's see here. Okay, Raquel Rivera, did I give you a part?
No, but my software is not working.
Okay, so I'll help you with that, Raquel. Just so that you have a part assigned to you. Raquel, let's get you started with the scapula, okay?
Okay, thank you.
Okay, Rochelle Heffelfinger. Rochelle, did you get a part yet?
I did not.
Okay, Rochelle, let's have you do the humerus and ulna as well.
Okay.
Okay. Sharia, did you get a part, Sharia? Sharia, did you get a part assigned?
Yes, I'm sorry. My computer was acting up. You gave me the occipital bone.
Okay, sounds like a good one. Okay, so does everybody have a part assigned at least? Okay. Now, obviously, gang, we're just getting started here. In truth, you know that you're each responsible for every part on this word bank, right? You must identify all of it. But for today, I want you to start with your assigned part. Learn what you can. Don't expect perfection. Don't expect perfection. But the idea is that we're going to learn parts and try to teach other parts as we go through the semester. Okay, that's the basic strategy here. So you all thanked me for the part I gave you. In about 2 weeks, you're going to be cursing me for the part I gave you. Okay, it's okay. I'm used to it. I know it's hard. I know it's challenging. Don't give up. It's okay to get frustrated. It's okay to need a break, but don't give up. Okay. So if you need help on some technology, please type "I need help" in the chat window. Okay. And there's no shame in that whatsoever, right? It is one of those darn things that happens. Okay, so I'm just making a list of folks that I need to help. And okay. So, okay, wow, okay. Okay, wow, my list is growing here. Okay, all right. So, here's what I want to do. I'm going to create breakout rooms like we did when I gave you an opportunity to talk with each other. I'm going to make sorry, I'm getting ahead of myself. We're getting short on time. So what I want to do now is a 20-minute private study session where you spend 20 minutes trying to learn the structures you've been assigned. That'll take us about to 11:50. At 11:50, we'll regroup. We will go into breakout rooms for 10, 12 minutes where you will share your beginning knowledge with each other, and then we will wrap for the day. So, to the folks that I need to talk to, I'm going to send you a link for a breakout room, pull you aside from everybody else so that you and I can talk together, okay? So if you don't need my help, you can go silent for a while. You can turn off your video camera. Just please stay alert when I announce that we're coming back together at 11:50. Go ahead and study. For the rest of you, just give me a second so that I can get that breakout room created. And I will be monitoring the chat window if you need some help. Okay, so just bear with me. Okay. So, okay.
Good morning.
Okay, folks. So I think right now I'll just stop the recording.